I can at least get through some of the preliminaries, and we'll get started here. So welcome, everyone. I'm Melissa Hanson, the Research Program Director for the Washington State Wine Commission. For many of you, you have attended some of our WAVEx webinars. If you are new, it's a joint effort between Washington State University and the Wine Commission, and it's research seminars and webinars to help push research information to the industry into the hands of users. And so we put these on annually. WAVEx is the shortened, condensed version of instead of a half-day research seminar, we just pick one topic and condense it and give it to you in bite-sized pieces. So today the focus is stuck fermentations. Dr. Thomas Henick-Kling has many years working on this, and this is a little bit different than just a research project that, you know, we're going to share results. These it's kind of lifeline learnings, and we have not been sponsoring specific research on stuck fermentations, but in the last couple of years, he has discovered some new discoveries about the glucose and fructose relationship that he'll be sharing today. So just as a quick reminder, be sure to ask your questions in the Q&A, and we are recording this, and everyone who registered will receive the link within about a day or so. And then I also will eventually be putting the recording link in the Wine Commission's website in the research library. One plug for some upcoming WAVEx webinars. Next month, Dr. Pete Jacoby is going to talk about his deep root zone irrigation work and some sensors that he's been working on. And then in April, Michelle Moyer will be talking about findings from nematode management, what she's learned about fumigation. All of these and the registration links are on the Wine Commission's website. And if you, under the research and innovation pages, there's a special WAVE section. And that concludes my part. So now I will stop sharing here in just a moment. There, okay, Thomas, it's yours.
I need to unmute myself too. You should be able to see my slides now.
Yes, you're good to go.
Perfect, yeah. Hello, everyone. Glad to be back, and my computer is behaving as we were hoping for. I apologize again for to those who last time tried to log in and we were not able to show the slides. Talking about sluggish and stuck fermentations, as Melissa said, it's something that probably most of you, all of you, have faced before. I did some research on this a long time ago, looking at nutrient requirements for yeast and bacteria, and then stumbled upon that we actually are very deficient in these musts. This was back on New York State, but it's the same here in Washington State. But a lot of work has been done on that over the years, and I'll summarize some of that for you. A few years back, it's almost 10 years back now, another cause of stuck fermentation. There we go. Oh, there we go. Another cause of stuck fermentations, the glucose-fructose imbalance, was discovered by a colleague of mine in Switzerland, Dr. Jörg Gaffner. And that's a cause that doesn't seem to be well known here in Washington State. I've seen cases over the years when I was back at Cornell, and we saw some recently here in Washington. So we thought it would be good to review the various causes and then tell you specifically about this glucose-fructose imbalance. So if we want a successful fermentation, we have to have microbes that grow well and function well. I have an example here of yeast and bacteria. no, sorry, it's just yeast. Non-Saccharomyces yeast need to grow. And then the Saccharomyces yeast, this is an example of a spontaneous fermentation. So we start at very low populations until we reach maximal populations where the fermentation is very active. So all these microbial cells need nutrients. We want to limit the nutrients initially there to avoid strong growth of unwanted yeast and bacteria in that early stage. And then we can manage the nutrients towards the end of the fermentation or after the end of the fermentation again. So here's my list of causes for stuck and sluggish fermentations. Nutrient deficiency is probably the major cause, whether you're doing spontaneous fermentations, meaning using the native population of yeast or bacteria, or whether you're doing inoculated fermentation with selected yeast and bacteria cultures. Temperature might be the next important factor. It can be too hot, it can be too cold. And probably most often temperature we run into trouble with is because of temperature shock. If the temperature in our fermenters changes too rapidly, go up or down. Alcohol, of course, is inhibitory to all cells at some point. We can have very high sugar concentrations in grapes in Washington, and that, of course, becomes inhibitory. There's competition among the microorganisms between various yeasts and various bacteria, and the yeast and the bacteria. Spray residues can be inhibitory to the yeast primarily, because remember, yeast are fungi. So any sprays we use to control powder mildew or botrytis will also have an effect on the yeast that we would like to use for our fermentations. At some point, the membranes of the yeast become leaky. They're lacking some of the important lipids that keep them fluid and keep them closed. And so that could be a cause of yeast dying and not fermenting anymore. And finally, I want to talk about this glucose-fructose imbalance. Yeast nutrition, when we talk about yeast nutrition, I just want to remind you, it's not just nitrogen. Of course, we need a carbon source. There's plenty of it in grape must. But besides the nitrogen, we also need a whole bunch of inorganic nutrients that I just listed here. The good thing about this is that all the complex nutrient mixes that are available from all the major suppliers of starter cultures and nutrient supplements contain sufficient amounts of these trace minerals as well. So it's a really easy way of protecting yourselves against potential deficiencies of some of these elements, which will also stop or slow fermentation. This is something very important to remember and know, and is that we talk about YAN and we talk about FAN. YAN is yeast assimilable nitrogen, and FAN is the free amino nitrogen that we can measure. So YAN is the sum of the free amino nitrogen. So that's the amino acids, the nitrogen that's bound on the amino acids. We measure how much of that nitrogen is available. That's typically the nitrogen that all microorganisms can use, yeast and bacteria. Grape must also contains ammonia, and yeast can take up ammonia and then use the nitrogen in there for making its own amino acids and proteins. So both are then measured in YAN, but they're two separate measures, two different chemical analysis that we need to do. There is an old method still around, the formal titration method, it works. We showed this some years ago. My friend and colleague Carl Scheibele did that work. It works well for measuring the amino nitrogen, but it uses formol, which is toxic chemical. You'd rather not have that in your lab. There's other methods for measuring the free amino nitrogen that can be purchased in ready-made kits. That's the way to go. Just to remind you, most wine microbiologists would say we need about 140, 150 milligrams minimum of YAN in our fermentations to get them to complete. Then the optimum rates or concentrations of YAN are quite varied, depending on how the tests were done, what other nutrients were present, what yeasts were used. So the range is actually quite big. It goes to 400 milligrams and sometimes even higher than that. We just had a seminar at WAVEx where we talked a lot about nitrogen nutrients in grape must, and reminded you then that our grape musts are typically very low in YAN, in yeast available nitrogen, much of it even below 100 milligrams, and sometimes as little as 50 or even 40 milligrams per liter. That was work that was done quite a while ago, published in 1996. So I looked for some more recent data, and I'll show those in a minute. To summarize again, we consider this a minimum here, around 140 milligrams per liter. Most people will go for something in that 250 milligrams per liter as an optimum that works well in just about all musts. But of course, the actual need, so if you're at that 250 milligrams per liter, you shouldn't be running into any problems, not even with different yeast strains and different temperatures and other nutrient situations. I mentioned the different methods. So the most common used one is this NOPA method that Christian Butzke published in 1998. That's the basis for various kits you can buy for FAN analysis, free amino nitrogen analysis. And here's some of the data that ETS labs kindly provided. This is just to let you know, yes, our musts are still basically as low in nitrogen as they were back in the 1980s. I have Cabernet Sauvignon, Merlot, Chardonnay, and Syrah. We have 5 different years from 2018 through 2022. In all of these musts, in all of these varieties, you can see there's quite a big range. It's almost a tenfold. Yeah, it's a tenfold difference from the lowest concentrations to the highest ones, 20 to 200. But the average typically falls, well, in Cabernet Sauvignon, below that 100, even more in that 50, 60 milligrams per liter of YAN. So that's the amino nitrogen and ammonia. In Syrah, it looks like it's a little bit higher, but still not sufficient for most fermentations. Chardonnay is probably the highest that we've seen lately, but it's still low. It might be just enough for many fermentations, but my question is, why would you do with just barely enough? It would make sense to supplement all of those as well. So that major cause of sluggish and slug fermentations that's due to nutrient deficiencies, we can today really eliminate completely by using by measuring the available yeast, available nitrogen, and adding yeast nutrients, and in some cases, bacteria nutrients as well. We also have better prepared starter cultures now that come preloaded with nutrients and reserve materials that they need to function for a long time. So that also is a great tool for avoiding any problems with stuck fermentations. Mistakes are still being made sometimes in preparing that starter culture so that you're injuring the yeast cells. And of course, the injured cells don't work very well and will get you into trouble. Temperature. Of course, there's a high temperature and a low temperature that is completely inhibitory or even lethal. Or yeast are quite adaptive, quite tolerant of various temperatures. The Saccharomyces yeast, the main fermentative yeast, they're happy at 18 to 35 or 16 to 35 Celsius. That's around 60 to about 95. But all of them will get damaged at temperatures over 95 or 100 Fahrenheit, especially when there's alcohol present, and in wine there usually is. So the combination of alcohol and high temperature will be lethal. So when you're running towards higher alcohol, you don't want to try to ferment at 90, 95 degrees Fahrenheit. The temperature is a great tool, though, to ensure that if you're using a native fermentation, native yeasts, you still want your main fermentation carried out with Saccharomyces. Make sure that temperature range quickly goes to between 18 and 30 or 35 Celsius. You're strongly encouraging native Saccharomyces yeast to do the job of fermentation in that case. Bacteria are not as tolerant to temperature, especially the ones we like to do the job of malolactic fermentation, Enococcus uni. They really stop metabolic activity at temperatures below 60 Fahrenheit or 15 Celsius. You have to really keep them in a temperature range of about 22 to 25. So that's in the 70s for them to do their job. And once it's done, of course, you cool the wine back down. So temperature, minimums and maximums are to be watched out for. Alcohol, I already mentioned, is a problem. There are quite a few Saccharomyces strains, so almost all of them are tolerant to 14 and 15% alcohol. And a few of the wine yeast will go to 16, maybe even 17% alcohol. But that's pushing it for most of them. And most of the malolactic bacteria don't do well over 14% alcohol. So it's better, and for flavor balance, it's probably better to keep that upper end of the alcohol close to 14%. There are a few other yeast and bacteria that are happy to get into environments that are over 14% alcohol. So you're actually, in a way, you're running a higher risk to select for spoilage microorganisms in these very high alcohol wines. Of course, non-Saccharomyces yeast, they are usually done, they're dead when the alcohol even goes much above 5%. High sugar concentration is another inhibitor. So say 26 Brix and higher is very inhibitory. There are some yeast that are more tolerant than others. What happens in these high sugar concentrated musts is that the high density of the sugar in the must actually draws water out of the cells of the yeast. The yeast dry up, they don't have enough water inside, and they they die, or certainly they're metabolically very inhibited. So there's not much you can do other than trying to get that Brix down before you run into problems, of course. Next, I want to talk about the microbial antagonism. I mentioned that already. We have various yeasts besides Saccharomyces, and the different Saccharomyces, actually, they will also compete against each other. So some of these will have produce specific chemicals, toxins that are targeted against their competitors in this environment. Of course, there are acetic acid bacteria and there are lactic acid bacteria that are competing, and especially acetic acid bacteria. And then these lactic acid bacteria, the lactobacilli and the pediococcus, all can inhibit alcoholic fermentation by inhibiting the sugar metabolism in Saccharomyces cerevisiae. Dr. Linda Bisson has shown that really nicely in some excellent work on the GAR prion mechanism. Enococcus uni very rarely is inhibitory to Saccharomyces yeast. It will be inhibitory to some other bacteria, but that's fine with us. These other bacteria we don't like to have in the wine anyway. But so the good thing is that Enococcus uni and Saccharomyces cerevisiae actually get along quite well. There's a competition for nutrients, but there's no toxicological inhibitory interaction between Enococcus and Saccharomyces. So that gives us the possibility to do this co-inoculation and have alcoholic fermentation and malolactic fermentation to happen at almost the same time. So that has its own advantages, of course. I mentioned spray residues already. Fungicides will also inhibit the yeast. There's a good, easy way to avoid that inhibition. If you suspect there is a fungicide residue, you can inoculate or treat your must with yeast hulls or yeast ghosts. Those are dead yeast cells that have been broken open. So it's the membrane in the yeast cell wall, and the membrane in the wall very effectively bind various toxins, including metals too, like copper. So they're a great sponge for binding and basically inactivating those inhibitors. And you can have a happy fermentation after that. And besides that, they're also providing some nutrients to the yeast and the bacteria. There's always some amino acids, some nucleic acids that are present there, and some fatty acids from the membrane. So you're actually adding a little bit of nutrients for the yeast and bacteria too. Lipid membrane deficiency, that really only is a problem if you're improperly prepared your starter culture, didn't watch that temperature of rehydration that's recommended. And of course, when we're running into very long and high alcohol content fermentations, the membranes of the yeast cells will become more and more deficient of lipids. The lipids are important to keep that barrier between the outside and the inside of the yeast, to keep that functioning. When there's a deficiency, that barrier, the membrane becomes leaky, and the yeast cell gets injured and eventually dies too because of that, the holes that are in its protective membrane. There are now also nutrient preparations available to help supplement, help the yeast to rebuild their cell membrane with some of these needed lipids. Finally, the glucose-fructose ratio. There are conditions which we don't fully understand where the yeast metabolizes the glucose very, very quickly and just doesn't get around to taking up the fructose. So a normal fermentation, we see glucose and fructose depleted to almost the same residual content. These are all dry fermentations where we would like to see them. And if you calculate glucose by fructose ratio here, you're sort of in that 3 unit. But we've seen grape musts here, and I saw them in New York State. People have seen them in other countries too. It's not a unique situation anywhere because we're all using the same yeast, really. And grape must with glucose and fructose in it. We can see these stuck fermentations where we have the glucose essentially depleted, but we still have a lot of fructose left. I've seen over 20 grams per liter of fructose and basically maybe a gram or less of glucose. Almost all of the starter cultures that we use today will not be able to finish this fermentation when we have this kind of imbalance. They're not able to take up the fructose that's remaining and metabolizing it. So there's 2 ways of, and here's the reference. This was actually published back in '93. Dr. Jörg Gaffner and Martin Schütz, his grad students, did that work and have done more work subsequently to trying to figure out what's causing that and how to fix it. So one way to fix this is obviously to use a yeast that can take up fructose. So it's a fructophilic yeast, and each of the yeast starter manufacturers, they have 1 or sometimes 2 yeasts that are fructophilic and can metabolize this. Another way to fix it, another easy way to fix it actually, is by just adding back some glucose or dextrose. And you can buy Baker's dextrose for very little money. So you could take these musts here and add, in this case, you would add about 10 grams of glucose. So you have a ratio of 10 to 16 grams here, and that would work. The same yeast that was fermenting there would start metabolizing again. The yeast actually, we learned, needs some glucose so that it can transport the fructose into the cell and metabolizing it. Now, you might not want to add more sugar to your must, even though this is a pretty small increase in alcohol. You don't want to increase the alcohol content. But, 10 grams per liter of sugar is maybe 0.1, 0.2% alcohol. So it's pretty minimal. So you have those 2 options. You can add glucose. You don't have to get 16 grams in here. Like I said, you can do it with 10. Here you would be fine with 6 or 8 grams of glucose, and that should take care of that fructose. Or you look specifically for a yeast strain that is fructophilic. You rack that wine that's stuck, get rid of the old yeast. It's not going to help you anymore, and re-inoculate with the new fructophilic yeast, following all the recommendations that the manufacturers have to restart, to help get this yeast acclimatized in this wine. There's a few more examples where this is also from Dr. Gaffner, showing the practical examples of stuck glucose fructose fermentations. In this case, he used a fructophilic yeast. This one is called 1895C. It by itself was able to restart these fermentations. Here we had 1 gram of glucose and 23 grams of fructose left. About 9 days later, both the glucose and the fructose were completely metabolized, and fermentation was done. Looks like they also had malolactic fermentation. Yeah, there's malate So the TA went down. Malate was metabolized at the same time by the bacteria. And there's some other examples. So that there's a fructophilic yeast will do the job very nicely. But you have to make sure you look for that yeast specifically. So this is just to remind you that these sugar analysis, they're really all enzymatic analysis that we have. Again, there are various suppliers of these kits for analysis. They use mostly the same chemistry or enzymology. Glucose is converted to glucose 6-phosphate. In the process, you get NADP reduced to NADPH, and you can measure that change with a spectrophotometer and then calculate back how much glucose you had. When you measure fructose, the fructose first gets converted to glucose 6-phosphate, and you do the same thing. So that's important. Don't want to bore anybody with chemistry or biochemistry here, but there are two separate enzymatic processes, so we can measure fructose and glucose separately, and that's something you have to be aware when you're measuring your residual sugar in your grape must or wine that has stuck fermenting. Make sure you measure glucose and fructose separately. Each of these analysis kits will tell you how you can get those two numbers. If you don't follow that separate analysis, you get just one number, which is the sum of all the fructose and all the glucose. And that won't tell you whether you have a stuck fermentation because of that glucose-fructose imbalance. Enzymatic glucose-fructose analysis has a very low detection limit, so it works great. You'll have to also respect that maximum limit. You have to make dilutions of wines that are stuck. I mean, 8 grams per liter is not much when you have 20 grams per liter of fructose in there. So be aware of the range in which these enzymes can work, but each kit will tell you what the range is, and you might have to make the dilutions. So I want to finish just by summarizing how to diagnose sluggish and stuck fermentations. It might start right at the beginning with a slow start. And that could be caused by temperature shock. When you prepared your starter culture, you were transferring that yeast culture too quickly from the 100-degree water to your must, which is maybe 70 or less than 70 degree Fahrenheit. So do that. Take your time to not shock the yeast with rapid temperature changes. The juice might be very clear. That's a good thing usually, but that might also make it harder for a fermentation to get started. Yeast hulls or yeast ghosts can be added, and that cures the problem. It gives the yeast and the bacteria basically a scaffold to attach, and so they stay in suspension in that liquid and start fermenting evenly. If it's very clear, the yeast that you add will just sink to the bottom, and it will take quite a while before they get enough metabolically active, produce enough CO2 so that they're lifted and get resuspended in there. So yeast hulls is a great tool to get the yeast dispersed and stay in dispersion in your grape must. It takes care of the toxins I already mentioned. It's never a good idea to go with low inoculation rates trying to save some money. It often doesn't work. Then you wasted all your time and the dollars, of course, for the culture that you bought and is not working now. Lack of nutrients is something we have to be always conscious of, and I'm sure you are, whether you're doing a native fermentation or added starter culture fermentations. We need to supplement our Washington grape musts. Sometimes the sluggishness or stuck happens after a few days of fermentation. Of course, we check daily to make sure we have a consistent utilization of sugar and the fermentation rate. We don't see any change in that brix in 48 hours. We would call that stuck again. This could be a nutrient situation, or maybe your temperature control on your tank went crazy or failed, or you already reached a very high alcohol content, or you might be running into a microbial population competition. We can measure, of course, nutrients. We can check the temperature. We can measure the alcohol. We can also have a quick look under the microscope to see what the microbial population is, or you send a sample out or do it in-house. You can plate for various yeast or use the PCR method to identify what yeast and bacteria you have and in what numbers. And that will tell you pretty clearly whether you have a microbial inhibition problem or whether it's one of the other problems. And then an easy one, finally, when you have a stuck fermentation, always measure glucose and fructose separately. Hopefully, they are still in about a 1-to-1 ratio. So you can rule this problem out and you look for the other causes. Just a few points on restarting alcoholic fermentations. You want to have it at a comfortable temperature, not too cold and not too warm. About 68, 70 is a good temperature, taking all the other inhibitory conditions in account. You want to get rid of the old starter culture by racking it as clean as possible. Those yeasts are no longer functioning and they'll not recover in most cases. They might also tell the next yeast you're throwing in, saying, hey, this is an uncomfortable place, and the new yeast won't even get started well. So best to get rid of the old yeast that got stuck. You can also use some materials that help removing some of these unwanted microorganisms. Chitin, there's various chitin preparations available. It's a protein that binds very effectively yeast and bacteria and helps settle them out and help inactivate them. Of course, you could do a filtration if you had to, really, maybe a cross-flow filtration or a centrifugation. If it's a bacterial problem, if it's lactic acid bacteria like Lactobacillus plantarum and Pediococcus, we have another tool, lysozyme, that will an enzyme that specifically kills lactic acid bacteria that can be used to remove that population, and then you can re-inoculate. I talked plenty, I think, about glucose fructose on how to restart that. Anytime you make a new starter culture or a stuck fermentation, you want to start that slowly and start by diluting the wine somewhat. In most cases, people recommend to start with half wine, half water, add some nutrients, and then add your starter culture, and then you slowly expand that once the fermentation gets going. And then you have an active starter culture that's well adapted to that wine, and that should finish the fermentation. Always monitoring, keeping an eye on that fermentation, the fermentation conditions, so that when things change too quickly or in a direction that you don't want to, you can intervene before you actually have a stuck fermentation. And then it's very important that you correctly identify the cause of this problem fermentation, because depending on what the cause is, there are different ways of correcting this. I think we have plenty of time left for questions and answers.
Yes, Thomas, we have a couple and I will give them to you.
Okay.
How about concentrations of fructose in the range of 2.5 grams per liter? Do you see them stick with that low a concentration of fructose?
Yeah, yes. When most wine fermentations actually will end up with basically less than half a gram of glucose, and they might still have 1-2 grams of fructose. I don't know if you call that a stock fermentation. A lot of winemakers will be okay with that residual sugar, but it's plenty of sugar for bacteria and yeast to start growing on, especially for bacteria. That's lots of sugar, 2 grams, 3 grams per liter.
Okay.
If you want to get rid of that, it's the same technique. You either add a tiny bit of glucose or you selectively add a fructophilic yeast. It's really your definition of how low alcohol, how little sugar you want. I call for myself in my wines, I want to see glucose and fructose below 1 gram per liter before I call them complete fermentations.
Okay, a couple questions here. What fungicides are truly known to cause stuck fermentations? Some studies, and some studies seem to imply that the quality of the yeast is more important than the fungicide residues. So what is your thought on that? And then can you distinguish the risk of fungicides based on white versus red wines? If you add the ghost yeast, will that alleviate this potential problem? So the first one is, do you know of specific fungicides that are known to cause stuck fermentations?
Well, all the fungicides that we use to control powdery mildew and botrytis will have an effect on our wine yeast, will be inhibitory to them.
So then the second part is, are there studies that imply the quality of the yeast is more important than the fungicide residues?
Sorry, say again, are there studies?
Can you overcome the fungicide issue with a better quality yeast?
No, because you can throw in a starter culture yeast and make it a sacrificial yeast. The first one that throws in gets poisoned by the fungicide residue and basically removes it from the wine, and then you re-inoculate and you're fine. But it's cheaper and easier to just use yeast ghosts or yeast hulls. When it comes to fungicides, there's one exception I'm just thinking of, copper. Of course, we're using copper in vineyard sprays quite a bit. Copper is universally toxic to microorganisms. Interesting enough, or not surprisingly, wine yeast have acquired quite a high tolerance to copper toxicity. They're able to remove that copper, move it into a form that's not toxic to them, because wine yeast have always been exposed, or for a very long time being exposed to copper and sulfur. So they have metabolic pathways to detoxify the copper and the sulfur, and they're fine with it. But the copper and sulfur will still be inhibitory to other yeast and to bacteria especially.
And on the fungicide issue, is there a difference between white versus red wines, or does that matter?
No, I can't think why that would make a difference. In fact, red wine might be a bit more dangerous because you have the skins in that fermentation for a long time. And of course, a lot of that fungicide is either on the outside or just inside the berry pulp. So there's a chance of having more and more release during the fermentation. So it's good precaution then to use yeast hulls to protect your active yeast.
Okay, another question is, can you comment on the differences in nutritional requirements between inoculated and native ferments?
Native ferments actually have quite a high nitrogen requirement because you're starting with a population that's very, very low, right? You're Saccharomyces and the non-Saccharomyces yeast, they're sort of in that 10 to the 2, a few hundred cells and less per milliliter. For an active fermentation, in all cases, you need 10 to the 6, 10 to the 7 to 10 to the 8, a million plus cells per milliliter. So that's it's a 1,000 or 10,000 fold growth or multiplication. That to build all that yeast cell mass, you need nitrogen and carbon and a few other nutrients. So a very poor nitrogen must will just not ferment properly on its own. But you can ferment it if you're throwing in a yeast starter culture, because you're starting at 1 million plus cells. So that yeast population only goes multiplies 2 or 3 times, and it has plenty of yeast. So it's very important to have sufficient nitrogen in for spontaneous fermentations. Otherwise, they'll be very drawn out, and you have all kinds of off-layers that might occur because you have things growing that you don't really want to grow, or the wine doesn't ferment for a while and you get oxidation problems.
Okay, and the last one, would you consider it better to manage your nutrition properly in the vineyard to the fruit coming into the winery with proper yan, as opposed to having to do it as an addition prior to ferment?
Yeah, it's a tempting thought, but it's not a good solution, because in order to get sufficient nitrogen into the must, which you can by putting more out into the vineyard, especially foliar applications are very effective. But you're creating problems in the vineyard. You're creating excessive vigor in the vineyard, so you have to spend more time getting rid of the extra shoots and extra leaves. You get bunch rot as a consequence very often of this dense canopy. You're throwing nitrogen on the soil when the plant doesn't use it. A lot of it goes up in the air. So you're contaminating your environment. You're throwing money away. I think it makes most sense to put nitrogen into the vineyard so that the plant is healthy and functions well, but is not overly vigorous. And also, you don't want to throw too much nitrogen on the soil that just gets lost. You're throwing money away. Follow the recommendations that we have on nitrogen management in the vineyard. I think we'll learn a lot pretty soon here, right, Melissa? Dr. Marcus Keller has a big study going here in Washington in our soil and climate conditions, looking at nitrogen management for several major cultivars, white and red, and we'll have finally much better guidance of what we use, when we use it, and how that affects the plant function and growth. And I know he's also looking with Jim Harbertson on what the yan and fan numbers are in that must. So we'll see how much we can improve those nitrogen content in the grapes without causing new problems in the vineyard. So that might be a couple of years away.
Yeah, it's entering its 4th year, so we're getting close to wrapping it up. But most of the nutrient recommendations were developed back in the 1960s in Thompson Seedless down in California, and so this is a big project. It's a federally funded, so it goes beyond just Washington. But because the Wine Commission is supporting some of his work, it started here, and we're getting data from our region as well. But it is a big project, so we're very excited to get some information out of this project soon. So does anybody have any other questions? That's all that got put into the, oh, one more. Do you really think wine yeast can withstand sulfur?
Yeah, it will make hydrogen sulfide. It uses some of that sulfur to make sulfur-containing amino acids too. But it's also a matter of the amounts, right? There's a limit of how much they can deal with, but they can deal with quite a bit, because over hundreds of years by now, they have been exposed to sulfur dust in the vineyard. But
So,
we don't really want the yeast to help to protect itself against excessive sulfur, because nobody wants hydrogen sulfide in their wines.
right. So that is it for the questions. If a question comes to you, feel free to, send it either directly to Thomas, or you can send it to me and I will forward it to Thomas. And we will have a link for this recording up within, like I said, maybe by tomorrow, as quick as I can. So that concludes this WAVEx, and please, this is my plea for you to take the survey. The link is in the chat box. Save it, and it will take you about 10 minutes. But it's really important to make sure we've got the right research priorities, and to make sure that there's nothing out there that's starting to brew and cause a problem that we're not on top of. So
Yeah,
with that, thank you,
if I can
Thomas.
just add, please don't hesitate to contact me if you have any questions. I love to hear them. I want to know if I was clear enough in what I explained. I want to know what's going on out there. Let me know what works and what doesn't work, so we can be helpful as we go along there.
Yeah, I might add, as one of Thomas's new roles, he's taking on sort of the enology extension role. So if you've got, application practice type questions, Thomas is the guy. So we look forward to beefing up that component of the V&E department. So
yeah, let us know what questions you have. I'll be working with Melissa and also with Washington Wine Growers Association to provide more seminars and workshops.
So great. So if you have topic ideas for future ones, send them to us. So with that, I will sign us off, and thank you. And we will have another one next month. The next couple ones deal on the vineyard side. Let me make sure I got, somebody didn't come up with, nope, we got all the questions. So, all righty. Thanks a lot, everyone.
Thank you. Bye-bye.